Welcome to Track 3, the Drug Development Pathway, sponsored by PRA Health Sciences. My name is Cassandra, and I'm excited to be hosting the Track 3 sessions. As a reminder, this track is being live streamed, and I would like to welcome our remote attendees, as well as remind you guys to download the mobile app, which is available, and we will be utilizing for the Q&A session today. This next session is laying the foundation, the role and goals of discovery and preclinical research, presented by Aris, Vice President of Research, and Randolph, Vice President of Drug Safety at Regeneron Pharmaceutical. Please join me in welcoming them.
Thank you, everyone. Can you hear me? I assume you can. I can hear myself. Okay, so I'm going to start off and then pass it to Randy. And there are really two things here that are interconnected, and I hope you'll see how. So let me see. Next slide. So I will touch first on the discovery research part, which is the one that's the most connected to what I like to call basic research. I don't make a differentiation between it, at least in our case, because of the way we work, although in other places it may be something somewhat different. And I should point out up front that discovery research and preclinical research share some important features because they take place mostly before a new potential, as we call it, candidate medicine, is tested in people. And they involve research that is done either in vitro, in test tubes and in cells, or, and I'm going to talk a lot about this, in animal model systems, what we call in vivo. So there's some terminology there for you. They do, however, have some somewhat different goals. That would be my focus, the discovery part. And what it does is it seeks to understand the basic biological mechanisms of disease. What is it that drives disease? What drives the phenotypes that are seen in patients? And in the case specifically for this audience, for genetically driven diseases, this also involves discovering what we call the causative gene and the variation in that gene that drives disease. Because we all have the same genes, and some of us, you have the variant that is non-causing, non-disease causing. In those of us who have disease, we have the disease-causing variant. And this is important. We call it genetic ascertainment, because if you can do that, then working out the key disease mechanisms that drive disease becomes easier, because you have more precise physiological models. You can create them so that they really reflect what is going on in the body of a human being with a disease. And if you can do all that, then it gives you a better handle at discovering drug candidates that may help the disease. And you also have a model, hopefully an animal model, where you can test those candidates for safety as well as efficacy. Preclinical research comes right after. And it seeks to understand these candidate medicines from the standpoint of safety. You want to know, are they likely to be safe in people? How they move through the body, what happens to them when you inject them. There is some technical terms here that you will hear later. And then decide on the kind of dose and formulation that is likely to work best. And that best part is somewhat relative in the sense that it's also about convenience, okay? Maybe you don't want to be injected with a drug every day, for example. So these are the kinds of things that Randy will touch upon. So what I'd like to do is break my talk in three parts. The parts are connected, and they have a sequence. In my mind, the sequence can occasionally be broken, can be done even backwards. But for the most part, in most cases, the sequence that I will present to you is logical and is the one that I find has paid off again and again. So in the case of a genetic disorder, you want to start with this ascertainment of causality. What is the gene or genes and the corresponding gene variants that are driving, that are causing this rare disease? And nowadays, this is done by sequencing. It used to be done by genetic mapping. It's been replaced by sequencing. And we heard this morning about whole genome sequencing, exome sequencing, or even doing genotyping on a panel. All of these things work depending on what the disease is. You may have to go to whole genome, you may not. A whole exome might be enough, or not even, because it is very possible to provide a successful diagnosis for many genetically different disorders just by using key symptoms. Something to remember. Sequencing is not an absolute must. But in the case of rare genetic disorders, sequencing provides a certain kind of certainty of what is going on. And the example that I have on top here is actually from a very large family. This is a pedigree. You can see in yellow here the affected individuals and unaffected. And by sequencing all of these, in this particular case, it's a specific example from work we've been doing at the RGC, we're able to ascertain the causative gene in an extremely rare genetic disorder. And we haven't published this yet, but hopefully it will be out soon. But most of the time, we don't have such huge families. This is, as you can see, a family with many children over many generations. What we have is something more like this, where you have trios or quartets, where you have the parents who are unaffected and the children who are, and sometimes a sibling where one is affected, the other is not. And it's by sequencing these different families that you can come down to causality. So if you look through the literature, as you look at more and more cases, you're going to be seeing a lot of examples that look more like this. That's kind of the norm. But after all this is done, and I should point out that there's one last point here. In my experience, through the work we've been doing at the Regeneron Genetics Center, this is absolutely key, that collaboration helps create insights in rare diseases. These are all different centers that see people with rare genetic disorders. We've been doing sequencing with them, and we've sequenced to date, I think, about 2,000 families, almost 2,000 families, about 15,000 people within those families, affected and unaffected. And you can see that we can find even novel candidate disease genes, about 300 or so. So this is something that really pays off. Once you have that, though, in order to get to what we call drug development, to enable drug development, there's an intermediate step. You have to understand the biology. If you don't understand the biology, the likelihood of creating a drug for any genetically different disorder, I would say, is pretty small. And the question that we're trying to address is, how do these rare disease-causing gene variants result in what is seen in patients? We call them phenotypes. There's also some other terms that are commonly used: mechanism of action, molecular mechanism, molecular pathophysiology. All of these are about how any given disease comes about from that genetic lesion, that genetic difference. And I've kind of picked through my own work for some examples to show you. I'm going to focus on one of them. But before I go there, I want to point out that, at least in my opinion, having a genetically accurate and hopefully physiologically relevant animal model of the disease can be an incredible help in deciphering its mechanisms. And earlier this morning, you might have heard about CRISPR. There was a question about CRISPR. But effectively, what we do is use different genetic technologies, either gene targeting or gene editing. It doesn't really matter, at least not in the mouse, to make animals, in this case is little mousies, where we have transferred the disease-causing gene into the corresponding place on the genome of a mouse. It can be done in rats, it can be done in pigs, depending on what you're trying to do. But mostly, the mice have been the workhorse for doing biology in the human rare disease space. And I will show you one example from my own work in a very rare genetic disorder. You heard about it this morning from Betsy. It's called fibrodysplasia ossificans progressiva. Because I have to say that to this day, from all the work that I've done across the rare disease space, it's been the one that has taught me the most, and still is teaching me, I would say, every day in the lab. Now, what I've put up here is two photographs. The one on the right is the skeleton of a person by the name of Harry Islak. On the left is a mouse model that we generated at Regeneron. It's not to scale. Mice with FOP are not as big as human beings. Don't worry, that's not a Franken mouse. The reason I put it as big is because I want to draw some parallels here in the structure or the phenotype, in this case, of that additional bone that forms in this disease. And you can see that it very well mirrors what is seen in this mouse model. And in some ways, one could say we were lucky because we work in the skeleton where the processes by which a skeleton is built are shared across vertebrates. It's a very well conserved system. But I'll take that. It's a nice place to be in. And what I want to take you through now is some important sort of milestones, and I'll try to make them simple without making them too, not getting too deep into the science, that taught us lessons that led us to find a drug candidate. And I think this can apply to many different rare genetic disorders as well. So this is kind of a case study as an illustration of what I aspire to every time I go down the path of yet another genetic disorder, to try to find a cure, or at least something that helps. It might not be a complete cure, at least something that helps. So again, this is Harry before he passed away. That's his skeleton. And I want to point out that what he did back then, it was in the 70s, to donate his skeleton to the Mutter Museum in Philadelphia was a very smart thing to do, because it was exactly this picture that got me interested in this. Here's our mouse, and the yellow here shows you the pseudo-colored additional heterotopic ossification, the production of this bone where it shouldn't belong. That is a hallmark and the major debilitating feature of this disease called FOP. Here's another one on the sternum. We haven't pseudo-colored that, but you can kind of see it. And having this mouse at hand enables now to start asking questions. Now, the first big discovery, I would say, and the one that enabled us to even make a mouse model, is that genetic ascertainment we were talking about. Finding out what's the causative gene. That was done by people at UPenn and at Oxford, and it was published back in 2006. And effectively what you're looking at is a receptor. It's called ACVR1, and we'll talk about what that means. What is a receptor? What is ACVR1? And this is a spectrum of mutations, of changes that are seen in this part of this receptor, on the inside part that sits on the inside of the cell. That is not so important. All I want to point out here is that this one variant, R206H, arginine changing position 206H being changed to histidine, is what we modeled in our mouse. It's also the one that's most common, and that's why we picked it. Now, in order for me to explain where we arrived at in the end, I need to give you a little bit of receptorology, as I call it. So what are receptors and how do they work? And more specifically, this one, this ACVR1. And I want you to think of them as kind of a switch. So a receptor is like a switch. And where it's engaged by something called a ligand, it goes from this off position, where the ligand has bound only part of the switch, to this on position, which has engaged the whole switch. So it's kind of like turning the lights on. Okay? Once this happens, a whole cascade of other events, kind of like electricity flowing through wires and turning on the lights or keeping your refrigerator on and so on and so forth, takes place. It's kind of like a molecular machine, if you like, that gets turned on by the sequence of events. The thing that was amazing, and if you can call it a lucky strike if anything is lucky about FOP, is that it turns out that this switch, this receptor, is what we call a druggable gene. It has some really interesting hallmarks that make it very tangible to go after from the standpoint of drug development. Not all genes are like that. That doesn't mean there is no way to go towards development of drugs. We could discuss that more later on. And in this particular case, the opportunities lie both on this outside part and this inside part of the receptor. I will not discuss these. These are being done by others. But the bottom line is that we can easily intervene either here or out here. And particularly in our case, what we wanted to know is whether the partner of the switch, what we call the ligand, was important for mechanism of disease, and if it was important, which one was it? Now, I'm not going to tell you all of this because that would be a scientific seminar that would be quite complicated, but the answer to that question turned out to be the key for the development of therapy. Now, in the rare disease space, I think you all appreciate the easiest thing to do is not to create a new drug. Yes, we do that. We try to do that when we can. But to reposition existing drugs. So both of these things, the development of new drugs and repositioning of existing drugs, is a game that we like to play. And this, again, I want you to remember, is enabled by knowing the genetic driver, the causality, the ascertainment that we talked about, but even more so, equally importantly, by understanding the biology. Because if you don't understand the biology, you can't ask the right questions. You can't ask what is important about disease progression so that you can actually get to this whole point of making therapies. Now, luckily, because of work that's been done over many, many years in many different companies, there's a whole toolbox of options that one can use. And in this particular example that I chose to discuss today, quite a few of them apply. So we talked about the fact that the inside of this receptor is an enzyme. It's a kinase. Kinases can be activated or inactivated by small molecule drugs. We have them in our arsenal. Some of them are approved products. JAK1 inhibitor, for example, that JAK1 is a kinase. So that's a kinase inhibitor that's in the clinic being used for rheumatoid arthritis. So small molecule drugs, these are typically synthetic, and they're usually administered orally, but not always. The other kind of thing which pertains more to what I do is what we call biologics. These are large molecules. They're typically proteins, but they can also be nucleic acids, modified nucleic acids, and so on and so forth. In our case, what we focus on primarily is antibodies or antibody-like molecules. But there's also other options, maybe not pertaining in the specific example here, but gene therapy, gene editing, as well as cell and tissue-based therapies. And again, you've heard about some of these. So what we focused on, though, was, again, on the outside. And to cut to the chase, what we found is that this abnormal bone that's made in fibrodysplasia ossificans progressiva requires activation of this mutant receptor by a ligand called activin A. Now, it doesn't really matter what activin A is. The bottom line here is that we discovered, by doing lots of different experiments, that in people who are not FOP, this receptor, this switch called ACVR1, works with ligands called BMPs to make bone. That's part of the normal building the normal skeleton. It turns out that activin also works in people with non-FOP, but it regulates this process by making a complex or sitting on the switch but not flipping it. So it's kind of like somebody holding the switch down and saying, do not turn this on. And you may wonder why is this, but it turns out that in biology, in every biological system one looks at, there are multiple mechanisms that regulate it. You want to turn it on, but you also want to be able to turn it off. And you can imagine what would happen if you couldn't turn something off. And FOP is kind of an example of this, where the dampener, okay, the brakes of the system, are perceived just like the gas pedal over here, and does the same thing. So this is a case, and I liken it to driving down a road and coming to a red light, and you know you have to press on the brake and stop your car. But in this case, when you press on the brake, if your car has FOP, you go through the red light at full speed because what the car sees is green, okay? And you can imagine, or you already seen, how catastrophic that is. But the bottom line is that this gave us now a handle that we could go after, because activin A is a protein that you can make antibodies to, and we were lucky enough that we had an antibody already from a different program. So this is a kind of example of what we call repositioning or repurposing. I don't particularly differentiate between the two terms, some people do. So we had this antibody from a different program, and then the question became very simple. Since we have a mouse model that gets FOP, can we put it in the mouse and stop FOP from happening? And I think it doesn't take too much to realize that if you do so, if you inhibit activin A with this antibody, the mouse is perfectly fine, whereas if you give it placebo, the mouse develops this heterotopic ossification. So this is, I think, an example that illustrates how starting from causality, genetic ascertainment, establishing causality, creating a mouse model, studying it to understand mechanism of action, you can figure out what you can hit in order to create a candidate therapeutic for this disease. And I think basically I just went through the summary, so we don't need to belabor the point. So I want to point out, though, that this is one example. In my lab alone, we touch a lot of different things. We're interested in rare diseases in general, I would say in a somewhat opportunistic way. If we can do something, if we think we can do something, we want to study the disease and get a better handling and have a yay or a nay, and then maybe we'll go after it. And I should point out that we work on lysosomal storage diseases. I've done work by proxy on spinal muscular atrophy by creating a mouse model or other contributing to it. Maybe some of you know about our work on cryopower-associated periodic syndromes and so on and so forth. So this is a list. It's not an all-inclusive list, but I want to effectively point out the research doesn't stop. It's not a one-disease thing for us. And the last thing I want to leave you with, and then I'm going to pass the baton on to Randy, is that if you were to ask me for a very opinionated piece of advice to you, as what can you do to help somebody like me, the researcher who wants to understand disease and hopefully find therapies or help even others find therapies, is to do these 5 things. And maybe it's repetitive because you've heard this before. A patient registry, preferably one. Imagine you want to find a restaurant in some town like Irvine, and there is a yellow book, a pink book, a green book, and a brown book. And none of them have the same addresses and the same restaurants. So you have to look at each one of these books and figure out which one can give me the address for the restaurant I want to go to. Well, that would be maddening. So it's the same for us. If we want to be able to reach the patients with a given disease, I don't want to have to go and talk to multiple people. And also, information will be captured differently. All of this, it just makes our lives what we do less efficient and our lives more difficult. Natural history. I mean, I can't stress enough how important having a natural history in place is. If you really want drug development to take place in the disease that you're interested in, you should do your utmost to get a natural history study done and do it in a way that is accessible in an equitable way. The foundation, the patient foundation, should own that natural history study. It should not be anybody else's. And the reason that is important is because this is the only way you can enable people to come in and start doing things for you. And by the way, this shouldn't be done anybody kind of a free-for-all. It should be done by using a scientific advisory board, just like with the biobank here. Okay, you want to have people who have no conflict of interest, scientists, physician scientists, patient advocates as well, regulate the access to the natural history as well as a biobank if you can establish one. Biomarkers, this is a very big topic. We don't have time for it. And they're usually a luxury, so I don't want to belabor this point. If you can get them, that's great. Not easy to get. And the last thing I want to point out, should you decide to engage the scientific community to help you, and I know you all do, that's why I'm here, and you want to do it by enabling their work, by funding it, you should highly consider doing it through a peer review process, again, one that is governed or overseen by an independent scientific advisory board with no apparent conflict of interest. So these are my thoughts based on my own experiences in the rare disease space for, I would say, over 20 years. Please.
Thanks, Aris. Am I on? Okay. Just want to make sure I got this right. Big green arrow advances, right? Okay, so I just want to build upon what Aris had talked about and sort of take you through, in a really short time period, a real high-level view of what is a real mix of disparate activities, but yet they're very integrated, and they're really positioned to move a molecule from the early progenitor stage to the point at which we feel we're comfortable with moving it to the clinic and actually exposing humans to this particular therapy. Therapies take many forms, obviously. So the goal of getting into the clinic, and the ultimate goal, obviously, is getting into the market or getting it to the patients in a way that we're beyond testing it and actually treating it as if it's a bona fide therapeutic. So, a little outline as far as what I want to run through in the next few minutes. Talk a little bit about who our stakeholders are, why we're actually doing this, and the reasoning behind it. A little bit about where we fit in terms of the R&D continuum, in terms of how you bring a therapeutic through its life. And then there are all these other things that are important components of moving a molecule from the very early stages of research to the point at which we can give them to people. So you have to make the stuff. You have to characterize it and be confident that whatever has been made is what you intended it to be and not something different or somehow a mix of other things. You need to formulate it in a way that people are able to take it, and I'll go into this in a little bit more depth, but basically put it into a dosage format that you can actually deliver it in a facile way. You need to figure out what's actually happening to the therapeutic once it's introduced into the body. So we use mammalian systems as models to figure out once it's in, what does it do, where does it go, how's it broken down, et cetera. One of the major drivers behind moving a drug forward or a therapy forward is basically having confidence around its safety. To feel confident enough to, feel confident enough to not be worried about introducing more risk than benefit. If all these things come together, the stars align, and you've got confidence, you're going to move into the clinic, and then we'll think about how we're going to introduce it into people. Something maybe a little less technical, but still just as important. We have to work with regulators to move the product through and ultimately get it to all the patients. And then I'll just close out with a little bit about, as Aris had said, what can you do to help us in our mission? So let's go a little bit further into the stakeholder list. It's very clear, who are our primary stakeholders. It's the people that are actually going to be the recipients of the drug, right? So it may be volunteers, maybe patients, but they're the ones that are going to drive the benefit, or at least help us understand the mechanism and the ability of the drug to do what it's intended to do. There are the doctors, right? So everybody knows that physicians are involved with this process as well. So it could be the physicians that are actually conducting clinical trials. It could be physicians that are thought leaders. They're the KOLs, the key opinion leaders, and so they're the advocates. They're the ones that are out and probably the thought leaders in the field in terms of understanding the disease and having access to patients. And then ultimately we've got the prescribers, right? And those, this may be a very blurred line between those three groups of physicians, but nevertheless, they're all very important in terms of our delivering information to them that makes them feel confident in giving this to their patients, right? So institutional review boards. So everybody knows that IRBs are there for a reason. They're there to make sure that what we're doing is reasonable and ethical, right? So this is all data-driven. They need to understand the profile of our medications, our therapeutics, to make sure that they feel good about allowing them to receive them. Government agencies. So, we've heard about the FDA, but it goes beyond that because we're developing therapies for the world, right? So FDA, PMDA in Japan, MHRA in Europe. There are a host of government agencies that we have to be involved with, work with closely, and partner with as part of our activities. Lastly, we have other types of partners, right? So, we may do this on our own, but more than likely we've got collaborators. So it could be on the academic side, and we're working with universities and other collaborators in that sort of domain. We may have people in the financial domain. So there may be a small company that has to deal with venture capital and investors and other people that are plowing money into the field and supporting their efforts. So, they too are partners in this. And also larger companies may actually partner with other large companies to offload risk. It may be because it's a very expensive endeavor. It could be that there's a certain uncertainty about the progress and the possibility of success, and so there might be some discussion around how best to sort of move it forward in a partnered way. And so all these groups, all these people are very important in terms of our ability to generate data and provide them with the information that they need to feel good about what we're doing and moving to the clinic. So The R&D continuum. So this is, maybe a little complex, maybe a little simplified, and you've seen elements of this in previous presentations. But what is clear is, basically we work within 3 primary dimensions. Safety being the most important. Safety is paramount. It tops the list no matter where you are. And you can see here, depicted in this figure, that it's sort of an iterative, but also a parallel process in terms of the various things that we do. So if you focus on safety first, you can see in the early stages of drug discovery and development, we do certain things, and as we mature the project, it goes through other elements of activity, and ultimately, hopefully to the end stage, right? And not just in safety assessments, but also medical utility. So efficacy is obviously part and parcel with safety, right? We're not going to deliver a drug that doesn't achieve the desired effect, right? So we want it to be safe, but we also want it to be effective. And so again, we're doing this iterative process in the efficacy situation to start out small, and it might be in the test tube, might be in an in vitro setting, moving into animal models of the disease, but ultimately we get into the model of choice, right? It's the patient that's suffering from the affliction. And so again, medical utility is the important second dimension that we work through. And thirdly, you have to make this stuff, right? So industrialization, that's maybe a very harsh term in a way, but I think it's realistic. We have to figure out a way to make this, but not just make it, but also make it in a way that's at quantities that we're not going to have a situation where we don't have enough to provide to the people that need it, right? So again, starting out very small and in modest ways, advancing through the development paradigm all the way to the point where we're mass producing it, at least to levels at which we can satisfy patient needs. So the R&D continuum. So let's get a little deeper into some of the various elements of preclinical research. So let's start out with synthesis and production. So can we make this therapeutic in quantity? So as I alluded to, we don't want to stock out, right? We need to conduct clinical trials. They could be small, they could be large, they could be many, they could be few, but nevertheless, we need to have the molecule to test in those trials in order to demonstrate the benefit and also evaluate the safety. But it goes beyond that, right? So we get on the marketplace, we need to be able to produce it in quantities that'll suffice to satisfy that market. Now, when we think about producing molecules, it really depends on the nature of that particular therapeutic agent. So as Aris had alluded to, it comes in many flavors, right? So we could have very small synthetic, small molecules that, think of yourselves as looking at a Tylenol or something that, that's in many people's minds maybe simplistic, but also, can be done in very simple terms in terms of its production. But then you might be getting a little bit more sophisticated when it comes to things beyond simple small molecules to making proteins or making viruses. And then you're expanding into much larger endeavors in terms of production. So now you're making substances by culturing cells to produce them for you. And so you may be in using engineering techniques and chemistry to have thousands and thousands of gallons and liters of culture medium that's producing your end product. But it could even go beyond proteins or viruses or growth factors or hormones. You could think of the endless possibilities, right? But we're coming to the age now where we're using whole cells, whether it's stem cells, or you may have actually heard recently about the approval of the CAR T cells that Novartis has come up with to treat a rare childhood form of cancer, which has great possibilities, but with it, it's a double-edged sword and also has great safety issues depending on how you look at it. So can we make this material, whatever that therapeutic is, in a way that is substantive and accurate and keeping in mind the various requirements that you may have to address based on what that particular therapeutic is in terms of its chemical nature or its organic nature. So we can make it in quantity. What's next? What do we do? Well, we have to make sure that whatever we're making, we're doing it in a consistent way. Can we reliably make this therapeutic? So we employ chemistry, we employ molecular biology, we employ all these different sciences. So, I don't want to regress, but there are a lot of people with very different types of expertises and experience that are involved with preclinical research. So chemists are going to be doing activities revolving around just demonstrating that what we produce is structurally accurate. Is it what we intended it to be? But it goes beyond that. It can't be just structurally accurate. It actually has to do what we think it's supposed to do, right? So there's this functional component to it. So we may be employing other assays, and this may take more of a biological flavor than a chemical flavor in terms of demonstrating that the drug, the therapeutic, is doing what it's supposed to. It goes beyond that. Potency comes into play, right? So it is what it is. It's supposed to be what it is. It is. It's basically functioning as it's supposed to, but is it functioning at the concentration that it's supposed to? So are we getting the effect at a certain concentration that we've seen in the past? Is it demonstrably similar to what we've experienced in the past? So potency is key as well, because that will relate to the dose that you give. So, consistency and uniformity. So we're not making a world supply from here to eternity, right, in one fell swoop. We're making batches and lots, and we're making stuff today that we may make again next week or next year. So we have to be confident that whatever we're making tomorrow is what it looked like yesterday, right? So there's this whole aspect of consistency and uniformity. And again, this comes into play in terms of making sure that we can give people confidence that what we're doing is not going to bring harm to people, right? Okay, so we can make it in quantity. We make it consistently. How do we give it? How do we give this therapeutic to people? Well, again, as I alluded to with the type of chemical or the type of therapeutic, there are certain ways of thinking about it. With formulation, you think about what the route of that administration might be, and that also is driven by the type of chemical, or therapeutic. So almost everybody's experienced taking, medicines by tablet or capsule, maybe even liquid. Your children in particular might have received drugs in that manner, so mostly small molecules are delivered in this form. But a lot of the compounds and therapeutics that we're working on cannot be given by mouth, and they have to be given in other forms. So injection is usually the most likely route, right? So it could be that you desire to get a drug or the therapy into the bloodstream in a very rapid manner, so it may be introduced into a vein. But maybe you want to introduce it in a slightly slower way and maybe in a blunted way, so maybe then it's given under the skin subcutaneously, or maybe it's a vaccine and you're getting it, in a big fat muscle somewhere to enable its activity. Then there may be other ways, right? So there may be specific diseases that you want to address at a more local level. So people can think of the various dermal diseases where maybe a topical approach really is the way that you have to go. And maybe it's a pulmonary disease, maybe it's asthma, whatever. And in that case, maybe you're looking at inhalation. There are a host of other things. Our company has a very important drug that's used for treatment of an eye disease, right? So the drug is actually introduced by injection into the back of the eye. So, and a lot of effort and work went into making sure that that particular formulation was both safe and effective. It sounds terrible to have to do that, but that's the way it's effective, and it's doing quite well. Overall, we figure out what we need, we create it, and then we want to make sure that whatever we've created is actually safe. It has to be tolerated, right? So we're not going to create something that's going to cause pain, that's going to cause damage. We want to make sure it's convenient to administer. So I think Iris had also alluded to this, that you want to make something that's actually going to allow for patient compliance. The worst thing to have is that you've created something and no one wants to take it because it's inconvenient, because of the way it has to be administered, or maybe it's painful, or maybe, for some other reason. And so that really defeats the whole purpose. So, we take that into account as we develop formulations. And maybe even more important, well, I shouldn't say more important, but just as important is the fact that whatever we create has to be rugged and stable. So, what can be given here in conditions in Southern California may not apply for West Africa. Your Ebola treatment in California may not be a big deal in terms of exposure to sun or heat or light, but maybe in West Africa it would. So, we have to think about where this medication is going, how it's going to be administered, how's it going to be used, how's it going to be stored. We want the stuff to be stable and rugged so that it survives and actually is going to produce the benefit that we desire. Okay, so we made it in quantity. We've proven that it's going to be consistent. We figured out a way to give it to people. So what's next? Well, we're going to introduce it into systems that we can actually characterize its behavior. So, there's this acronym that we call ADME, and you can see the letters a little bit further down on the slide as far as what those things stand for. So, basically this is a series of studies in animals that basically is going to provide us with information about how the drug behaves once it's introduced. So the absorption of the drug, how is it taken up into the body? And that may vary depending on how you give it, right? So orally, it may be a little variable. Intravenously, you may have a very high spike of drug early on, and then a washout. Maybe you give it in another manner, subcutaneously, and maybe it's a little different, a little less of a peak and a little more blunted. So these are all the things that you have to consider in terms of the possibilities. So this is something that you characterize in an animal model system so that you can then do some predictions as far as how it might behave in the human system. So, once the drug's absorbed, then you have to figure out where it's going to go. There may be a desired place for it to go. Maybe you don't want it to, target any specific spot. So we do studies to actually examine where does the drug concentrate and where does it go. Okay, it's there somewhere. how does it go away, right? So there's the whole concept around metabolism, catabolism. The drug is going to break down into something else at some point because of natural, biological, and chemical processes. Is it going to break down into something that's inactive? Is it going to break down into something that's toxic and cause problems? Is it going to affect a metabolic pathway that's going to affect another drug you're taking? So, many of you are probably aware of, co-medications and that there are risks for drug interactions. So this is the kind of work that we do to understand how a therapy might actually go about affecting a metabolic pathway that may interact with another medication that you're taking. Polypharmacy is a very common thing these days. It may also make you think about whether, how you take it affects how it's broken down, or how it's absorbed. Food might be an impact. Do you take it on an empty stomach? Do you take it on a full stomach? Do you drink grapefruit juice with it? Which is another big issue in certain drugs. Okay, so it's going to break down, but then it has to be eliminated, right? So how long does it stay? How does it leave the body? Does it go through the liver and out the feces? Does it go through the kidneys and out through the urine? Does it get exhaled? Alcohol, you blow off a certain amount of alcohol through your breath, right? So we need to understand all these elements of drug disposition so we can better predict what we think will happen once the drug is introduced into the clinic. And so by and large, this is done by chemical analysis of both bodily fluids and tissues. And as I said, this is really critical in terms of our ability to predict what might happen in people. Okay, we made it. We've characterized it. We formulated it. We figured out at least what we think is going to happen in the body. Then we've got another big ticket item. We have to talk about safety. So safety in terms of using various tools, and these could be in silico, so computational tools, computer tools, in vitro tools that are more like test tubes and plates, in vivo tools, animal models. In cerebro, I always put that in there a little bit jokingly, but I think there's a lot of thought that goes behind some of this. So, we're using our heads as part of this evaluation. And so what we're trying to do is assess the potential for adverse effects, hair loss, adverse effect, right? Undesirable effect, hair growth. Well, depending on the person, I guess, it could be undesirable or desirable, right? So, but that's sort of the thing that is important to sort of divine out because it's going to play into how we administer the drug, whether we do or not, and how we're going to dose it, right? So, we're employing a lot of very basic techniques in terms of toxicology and pathology, both on the veterinary and the human side. And the basic kinds of testing that are done are very similar to what you do for a human physical examination. So, there are specific observations and measurements that are done. Blood is taken, serum is taken, urine is taken, et cetera, et cetera. Electrocardiograms, you can think of the whole host of things that you undergo as part of a routine physical examination. The only thing that we add to that is a post-mortem, and so that's not part of a routine physical. But it's a very complete picture that we can draw from a very complex and integrated set of studies that we do. But it goes beyond just general homeostasis and well-being. We also do specific studies that revolve around whether a medication or a therapeutic is going to affect fertility or ability to reproduce. Is there an impact on the developing fetus? These are all important questions to ask. We may also want to think about whether the drug or therapeutic has a direct impact on DNA. Are we causing cancer as we treat cancer? It's something we need to keep in mind. So, now we have a drug that we've made in quantity, we know pretty well, we can formulate it, we know how it's going to behave, we think we know whether it's going to have a liability as far as safety. What's next? Well, maybe now we've actually got a body of information that we can keep together in a way that will give us the confidence to actually, say, yes, it's time to move into the clinic. So, if we've decided that, then we've got to integrate all this information, right? So we've got a bunch of dots that we need to connect. And so we've got the pharmacodynamic data, right? So we've got the activity information, the efficacy information. So whether it's in vitro or out of Aris's model for FOP. We've got information on pharmacokinetics, the disposition of the drug. So we know how it goes in, we know what happens when it's inside, we know how it leaves. And then we've got the safety information. All these data have to be integrated in a way such that we arrive at a number, a way of giving this medication in what we would hope to be a safe way, a potentially efficacious way, and a convenient way that actually will allow us to initially evaluate the drug or the therapy in the clinic in what could be a volunteer population, or it could be appropriate patients. It depends, right? So we need to connect all those dots. But it's clear that once you enter into the clinic, you're at a very formative stage and you're asking very basic questions about safety, maybe how the drug is behaving in terms of its disposition, its ADME, and hopefully you have a biomarker or some other readout that you're actually going to glean some information about its potential efficacy. But it's just the first step, and that next step is obviously much more robust and rigorous in terms of larger and larger clinical trials. Obviously, rare diseases might have a slightly different approach, but nevertheless, this sort of, I think, in a general way is maintained in terms of how things go. So first in human testing is important in terms of its ability to establish a safe starting place, but also divine out information that would be important to building on so that we can get into the clinic and actually build out a really strong efficacy and safety data set. So we can't forget the regulatory piece, right? So a lot of that other stuff is technical and scientific, not to say that our regulatory colleagues are not that way as well, but they're focusing on maybe some other things as well, right? So we cannot go anywhere into the clinic without engagement. They are the gatekeepers in terms of entry and also approval, right? So it just behooves us to set up really good relationships with our regulators in terms of having very early contact, frequent contact. This is not a relationship that should be passive, but it should be active, and we shouldn't be dismissive. We should be very receptive and really have good dialogue. We want to be partners with these people, with these agencies. We want to make sure that they're involved and really integrated into our thinking in terms of planning and initiating and actually conduct of clinical trials, because ultimately the goal, as is for everybody, is to get an approval for a desired indication with a label that will actually allow us to treat the patients that we need to in a way that's appropriate. So, my recognition is that FDA ain't Japan, it's not Europe, it's not other countries, right? Every regulatory agency has a different set of experience. They may rely on each other for certain things, but by and large, they have slightly different ways of thinking. So when we think about interactions, we have to think in a more global fashion, because we know that some remote region may not have the sophistication or the experience that maybe our own US regulators may have. So we want to make sure that we're doing this in an appropriate way so that we can get the best out of the interaction. So lastly, how can you help? So I think you've kind of gotten the gist that animals are a big deal for us in terms of our ability to divine out biology, figure out safety, and a lot of other things that are really important to putting together a drug submission, right? So they're our key and well-proven tool in our kit. We want to make sure that we've got good access to them, right? The work that we do with animals is very rigorously overseen and also conducted, right? So just as we have IRBs for clinical trials, we have something called the IACUC, the Institutional Animal Care and Use Committee, for oversight of our protocols and review. So everything actually that's done in an animal actually has to go through this review. So it's very akin to an IRB. Saying that, although animal models are really key to our work, we're always looking for ways to reduce, refine, and replace these animals in our work. And when we can find alternatives, we will employ them, as long as it's in keeping with what we're required to do as far as regulatory requirements. So, it's an important part of what we want to point out is that, we're just not dogmatically using animals as part of our work, but really using them thoughtfully and also with respect. So this is where you come in. So, this is something that sort of comes and goes depending on the geography of the world and the date, but we are often challenged by extremism in terms of animal rights activities, and there's definitely clear movement in certain areas of the world where they've been successfully able to actually prevent or inhibit pretty drastically our ability to maneuver in this field. So our plea to you is that if you encounter this kinds of extremist views, I think the things that we're laying out here in terms of our ability to do things in a very thoughtful and respectful way, and in a way that's very much ethical, with the oversight of IACUC, should really allow us to have freedom to operate here. We're not here to do things in an unethical way, and that we really need to have the freedom to operate. And so any advocacy that you can provide in this regard would really be helpful. So I'm just going to take it full circle now. So conclusion. So just to reiterate, like ARIS in the discovery space, preclinical research is important. It's an integral component of what we do in terms of moving successful therapies through. While our discovery colleagues are understanding very basic biology and genetic drivers, the preclinical research is often done to kind of flesh out the understanding and the profiles for those drugs so that we can move them into the clinic. So we definitely want to produce these drugs in a way that's consistent, substantial. We want to be able to characterize them in a way that we can prove that they are what they're supposed to be. that we can track them and ensure that they're safe, right? So we also must be able to formulate them in a stable fashion so that they can be given consistently and conveniently, and also that the regimen that we've divined out for the clinic is one in which we're giving the right drug to the right patient at the right time, because that's key, right? So also regulators are our partners, and we want to engage them in a meaningful way, and frequently often is better than never, or not so often, and any help that we can give in terms of animal models and our support in terms of having freedom to operate there is obviously much appreciated. So I hope I've, in these fast flash few minutes of talking, that I've actually given you a little bit of a greater appreciation for the fact that it takes a village to do this work, and that no one person or group can actually do all the various things that are required to move a drug from its creation through its preclinical development into clinical development. And so that it really takes a village, and we're all part of this together. So thank you for your time, and I think we'll stop there and see if there are any questions. Sandra, leave that there for you.
Thank you both very much. So we do have a few minutes to answer questions, so either feed them in via the app or there are mics up here. Go ahead.
Hi, that was wonderful. Thank you. You mentioned the need to have one registry to go to get your data, and I'm just wondering if your experience has been that in order to find sufficient data for one specific disease, that you find yourself looking at 3, 4, 5 registries to find different aspects of people's experience with that disease.
Well, I'm not going to name any names, but I've had experiences where I'm trying to get to something, and I find that the landscape is fragmented. And not only fragmented, but there's animosity between different factions. That really doesn't help your cause. I don't have a solution for it, but at least if you're starting from scratch, if you're starting de novo, if you have no legacy, so to speak, right? I think it would pay off to think about having one registry and one only.
All right, thank you.
All right, well, thank you both very much for the great information surrounding preclinical research. At this time, if you can all jump onto the mobile app and take the brief survey. This does conclude this session, and we can head out for lunch. It will be served outside on the lawn by the pavilion and is sponsored generously by Shire. So enjoy the sun and your delicious lunch. Thank you again.
I don't wanna know that, babe.